uh, to discuss papers. Okay, let's
Yeah.
start. So now we have
Okay,
frogs.
so
Greek
this is
frogs. Let's go.
not only, yeah, not only Greek. There are, you can
Oh,
find them in the temperate zones of the Europe. So I was telling you before that this is a work we have done with Vasilis. Vasilis was my professor in my as a PhD student and master when I and a master student also. So this work was a field study of this species, this frog species, Pelophylax ridibundus. I think that it had a former name, like it was Rana ridibunda, but it was renamed afterwards. So when we started working on this animal species, it was called Rana ridibunda. And while we were working and we revised the first or the second paper, one reviewer told us that you have to change the name because it says it's changed now and it's called Pelophylax ridibundus. So if we go to the next screen uh, and press again, yes, we can find. Okay, so this work has been published in two different papers. The one in Comparative Biochemistry and Physiology, Part A, in 2012, and the next year, the supplementary results mostly about the apoptotic death and oxidative stress were published in cryobiology. So if we can see some things about this frog, about this animal species that we have investigated, if we can go to the next slide.
Costa, I
Uh,
used this
yeah.
2013 paper in my class.
Oh, okay. That's really really
In
nice. Thank you.
2013. Yeah.
It was yes, it was in 2013. Yes. So you know
I
this
used
work
it in
partially.
my classes in 2013. That's nice.
Okay,
Okay.
that's an honor.
Okay.
Okay, so can we go to the next slide, please? And press again and again. So this is the, yeah, okay. So we can find some information here. This is the frog that we were working about. This was a field study. You can see the classification and then you can see the map. So you can see that it almost ranges in the temperate countries in the Mediterranean and some in some Mediterranean countries. And you can see that in Greece, it's usually located in the northern part of Greece. We know that most amphibians, and this one, Pelophylax ridibundus, go under hibernation. When we can go to the next slide, Marina, and press again and again. Yeah. So, oh no, 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 just before. So we know some things. We know all of us. We know that when we go into torpor, the metabolic rate can be really, really small compared to the normal. And usually in complete hibernation in torpor, we have a ventilation of really, really low level, like 1 or 2 breaths per minute in small vertebrates, so that energy stores can last longer. If we see here this graph, we can see that these dotted arrows indicate some preliminary trials. So we know now that animals go into torpor like in a smooth way, not like straight into torpor, but it seems that like that the animal prepares to go into torpor. And then when it activates from hibernation, during arousal from hibernation, we have something similar also. And we know now, if we go to the next slide, that during this period, you can yes, you can appear at all. So we know that when we go into torpor for these amphibian species, it goes into hibernation due to temperature decreases, or due to low oxygen pressure, or the lack of food. Okay, during winter, when in the north hemisphere, when this animal species goes into torpor, we have biochemical and physiological changes. And these have to do with a marked decrease in the oxygen consumption and metabolic rate during the torpor. But if we see this orange the orange shape during the arousal, we can see that if you can press, Marina, the next buttons, we see that during recovery we have this activation and this arousal and this aerobic capacity increase due to the mitochondria, because we need ATP to be active. But this activation causes oxidative stress, and this oxidative stress, due to the imbalance between ROS production and the antioxidant defense system, can result to some damages in cell substances like proteins, nucleic acids, carbohydrates, and lipids. You can press, Marina, the next button. Okay, so if we go to the next slide, again, if we go to the next slide, we can see that due to the oxidative stress, which comes from the arousal, from hibernation, the literature has presented some of cell protective processes, like the heat shock response, which is induced under oxidative stress. Also, parallel to this heat shock response, we know that the antioxidant defense is also increased in order to cope a bit with the oxidative stress produced during arousal. And also the apoptotic mechanism, the apoptotic cell death, is also induced because oxidative stress goes hand to hand with the apoptotic cell death pathway. So these are the pathways that we have aimed to investigate in both of these studies. So if we can go to the next slide, we see that we have taken this species, this animal species, and we have acclimatized it during a whole year. And we have taken a monthly sampling, or nearly a monthly sampling of the heart of this frog, and then we have examined the processes and the mechanism, the cellular mechanism I was telling you about in the previous slide. We have to tell here that except from the heart, we have also taken the muscle, the gastrocnemius muscle. But it was, I thought that if I also included the gastrocnemius muscle results, it would be a huge presentation and it would lead to the same thing. So I have decided to include only the heart in order for this to be more simple and focused. So can we go to the next slide, please? And so now, concerning the results, we have also tried to examine HIF, hypoxia-induced factor. So this was a really difficult protein for us at the laboratory of Vasilis to cope with, but we managed after all, and then we got really good results on this protein. And we can see that these levels, the degraded and non-induced levels of this hypoxia-induced factor, showed the lowest values not only during arousal, but also during overwintering. And this tells us that both during arousal and during overwintering, the torpor, there is oxidative stress. And here in the slides, we can see that overwintering is colored with blue, and then the arousal from overwintering during the next months is shaped with orange. So these results concerning the HIF-1A factor has shown us that under both these periods, torpor and arousal from overwintering, we have oxidative stress. If you press next, Marina, we will see this result. Okay, and if we go to the next slide and press again, we can see also that this was also the fact for the antioxidant defense as well, because we can see increased levels. Okay, most of them are mostly increased during arousal, but we can see similarly increased levels both during overwintering and both during arousal. So this started to give us the idea that oxidative stress in this animal during this whole period of overwintering and arousal comes not only during awakening from torpor, but also during overwintering. And we can see also the next slides. If we can go to the next slides and we can see the heat shock response, we can see that at first, well, the HSP70 member is mainly induced during arousal. But if we see HSP90, we can see that its levels are induced, the most increased induced levels of HSP90 can be seen also during overwintering. So we know that the cell needs some protection, okay, also during overwintering. So something is happening, probably because we have seen from the previous results that oxidative stress is not only happening during arousal, but also during overwintering. And then if we can go to the next results and we can see the apoptotic death. Oh no, the map case, okay. The map case saw us a bit different pattern. Mostly they are induced during arousal. Okay, we can see also some increased levels in some MAPK members like the ERGs, the activation of ERGs during overwintering, but we can see that they are all increased, these activation levels, during arousal from hibernation. And if we can go to the next one, to the next, okay, so here we see the apoptotic cell death, and here we can see that the pro-apoptotic members like BAX, okay, BAX is a pro-apoptotic protein, like the one which induces the apoptotic cell death, is increased mainly during overwintering. And we can see that when we go to the arousal from overwintering, we can see then increased levels of the BCL family, which is the anti-apoptotic. This comes to stop the apoptotic cell death induced by BAX. And we can see also increased levels of the ubiquitin conjugates, which play a very versatile role both in apoptosis and also during autophagy as well. So if we go to the next slide, we can see here, okay, that all these members of these cellular pathways that we have examined is that they are located differently, some of them differently and some of them together during these two distinct periods, the one of overwintering and the one of arousal from torpor. We can see that during overwintering, we have BAX, okay, the pro-apoptotic member, which induces the cell death, but during the arousal from overwintering, we have some other distinct members of these cellular pathways, like the HSP70, MAPK, okay, BCL, the anti-apoptotic member, and ubiquitin. And in both of these periods, we have distinct induction of hydroxy-HIF-1A. We have the antioxidant defense, which is increased during both these periods, but we see a slight increase, like when we're going to the arousal, okay, of this period, and we have also the HSP90, which is present in both of these periods. So, do we discuss about pause here? Is this preparation for oxidative stress strategy? When does it happen? Does it happen during overwintering? Does it happen during arousal? Or does it happen, okay, because we see that oxidative stress is present in both of these periods. Do we have to see what's happening a bit before the animal comes into the overwintering process? Okay, so that's why I have this question mark in this green shape. Like, it would be really nice to see what's happening in a few days before the overwintering. However, if we go to the next slide, we can see that these all cellular processes have they come in queue in the cell. So we know that the oxidative stress can induce the HSF monomers via the MAPK pathway, and then these monomers, which are phosphorylated, they go into the nucleus and they activate the HSE elements, and then the HSP's proteins 70 and 90 are induced in order to repair some damage done in the cell. But we can see that oxidative stress can also induce some other elements in the DNA, and then the enzymatic antioxidant defense can be increased, or the apoptotic cell death pathway can be increased. And we have here the pro-apoptosis and anti-apoptosis, like a tug of war, the one going to induce apoptosis and the other one going to decrease apoptosis. But we see that after all, and all this oxidative stress and all these cellular events happening in the cell, they all come to one point, and they all have one aim, like the cell survival. And in fact, we didn't have any cell deaths, any animal deaths during this experimental procedure. But if I would do this thing again, this whole experimental procedure again, I would schedule something like a different, I don't know, a different protocol, like being on days prior to hibernation. And I don't know if I could take every day some specimens, or every 3 days, or every 5 days, but it would be really nice to see what's happening before. So is there a preparation for oxidative strategy before overwintering? These are my frogs. These are Vasilis' frogs and my frogs. And it was really nice for me to having presenting you this work done about a decade ago in the lab of Vasilis. And I hope you enjoyed it. And I hope that we can discuss a lot about these frogs today.
It's impossible to figure out at the molecular level what was going on in 2013. But a revisitation of this paper in using what we know today, it's already very interesting.
Mm-hmm.
You see the idea that of that project we started to talk about on HSPs and
about all the things that we were saying in our previous seminars.
In our talk,
and also in our talk.
We could use this paper and re-evaluate, using what we know today, because these results are 11 years old. So we didn't know many things back then. But the data are the data. The data are forever.
They exist. They are forever.
The data is forever.
It's written, never deleted.
But the interpretation can change.
Of course.
Okay, several species. We start with frogs. And we simulated the natural field conditions in the lab. But it works really perfectly. I mean, we got nice results about this hypothesis. But it was your idea about this experiment. You hear me?
Yes, we can hear you.
Oh, sorry, because I'm seeing experiments. Okay. Presentation. Okay. And really, my point, Marcelo, on this post-hypothesis is that, okay, we see that we have the expression of some enzymes, antioxidant, enzymes, salt, catalase, etc. But I think that the most important is to see, to associate these results with energetics. How it does work with energy demand during hibernation, during hypoxia. And of course, I'm sorry, just I'm asking again. Can you hear me?
Yeah, you want to associate with what?
With energetics, with the metabolism.
Energetic. Yes, yes, yeah. If you're using your
That's
snail
the
hypothesis.
most important, because the oxidant defense isn't only the mechanism that involved in the adaptation of these animals. And that is the big question. To be able to connect, to associate all this data with energetics, what's happening during hibernation, recovery, etc. We know that, okay, we have a Z expression, but what about the translation? Because it's energy demanding processes, all these processes, physiological processes. And I think that, and of course, it's tissue specific. All these strategies are tissue specific.
Yeah.
And, okay. And really appreciate a lot your effort to discuss all this data and all these people involved in this group to see from examining several species to see what's happening under several environmental conditions, whether this is a universal hypothesis or not. Okay, this is, I don't know, that's my point.
Yeah, I just have one question. How do you maintain these animals? They're in water. You make a like they burrow?
Very important question. Yeah, it was a very important question. Just we let the animals to behave as in the nature, as in the field. Okay.
No, I think Marcelo is asking how did we maintain these animals in the field? Are you
Okay.
asking that, Marcelo?
Okay.
Yeah, that's it. Yeah,
It was
if they're
it was
in the water, if they're the burrow.
They were in open aquariums. Okay, there were exposed these aquariums in the field. They were covered, and then we had soil in them, and then we had water. So it was something like a small pond inside the aquarium. So when they wanted to burrow inside the soil, they could do it. And when they wanted to go out into the water, they could do it also.
Do they get hypoxic during hibernation or they not? They do not.
They get a hypoxic because we see this from the hydroxy HIF results. And we see this from all the cellular responses during overwintering. So we know
but
now that
HIF goes down during hibernation. Yeah.
So no, this was so we couldn't find an antibody at this time of when we did the experiments, and we couldn't purchase an antibody. So I think that we found someone who created an antibody, and we sent an email, and we didn't get an answer. So we find this hydroxy HIF. So this is a form. And when the hydroxy form is down, we know that HIF is activated. So if you see the levels of hydroxy HIF going down, that HIF is activated. So it's something like a different, an opposite response.
Okay. If hydroxy is down, HIF is
yes.
Is up.
So the active, yes. So if you have hydroxy HIF down, you have the active form of HIF up.
Okay, I got it. Yeah, but my question: they go in a burrow, so the burrow creates a hypoxic environment, or they breathe less and they become hypoxic? Because these are important questions to understand the natural biology of these animals. For example, do you remember Daniel's presentation, his frogs? So they do a burrow, they can go down 2 meters, but they do not go hypoxic. They depress metabolism, but oxygen is there is plenty of oxygen in the burrow because the sand is very granulated. There's big grains, so oxygen goes reaches these animals, goes under in the burrow. So
okay. So these animals also, for when they go into torpor, they have to burrow. They have to go into the sun. They have to go into the soil. So when they overwinter, they have to be inside the ground. I don't know, but we didn't have anything to measure if the ground gets hypoxic or if oxygen could go into. But I think that in nature also they do not burrow in such deep, such a depth like 2 meters. They go really.
But depending on the kind of material. Yeah, if it's a mud, certain kind of mud is it has problem with oxygen. Yeah, to deliver to the animals, so they get hypoxic. Certain kind of sand, it's very fine, so it always has a problem. So when we go to nature, it's very important to understand the location. For example, I want to invite Professor Debas. He's from India. I think he's not here today. He's done very nice experiments with toads from India they're in caves, in hibernation. And so, but I don't know how is the environment in his case. So it's very important to understand the environment so we can get the abiotic stressors, the factors, exactly what is going on. Because in the past we were, I mean, everybody of us, we were more interested in the biochemistry. So we didn't think very much about environment. But if we want to see the mechanism in nature, we have to understand how are the abiotic factors, which abiotic factors are influencing the animal. So we have to have a more ecological look to do this kind of work. So I do some jokes with my colleagues. I say I tell them now I'm not about biochemistry anymore. I'm ecologist. So you want to go to this path.
You are right, Marcelo. You are right, really. But it's very difficult to simulate all these conditions. Okay, it's very in the lab. Okay, that's why it's be very nice if you have a field stations and to see what's happening over there. Okay.
We can collaborate with this, and we have friends that they know much more than us. Friends from São Paulo. They are true physiologists from that work in nature. They don't care much about biochemistry, but they care a lot about the nature itself. So it's nice when we collaborate with the zoologists. So we get a bigger picture of the relationship between the animal and the environment. So only with all this information, we will understand what's happening. This is what I think.
You're right.
We have to marry these things together. But I was thinking that it goes into torpor, it goes into hibernation, so it becomes. This is a question, more of a question, like I'm trying to understand. So I'm a frog and I'm going into hibernation. Okay, so I'm becoming hypoxic.
The thing is, Yeah, because of your HIF data, you think they are hypoxic. It's an indication.
No. This is a question. I'm asking Costas because I want to know some things. So,
Okay.
and I want to straight some things in my mind. So, I am a frog and I'm going into torpor. So this means that I have metabolic depression. I don't breathe because I have to cut on the oxygen that I breathe. So I become a hypoxic. Otherwise, or not.
So it depends. Again, for example, the frogs from Daniel, they don't get hypoxia. They're fine. We didn't measure oxygen in the blood, but from the data, we are sure that they're normal oxygen, even though they are in deep metabolic depression.
Yeah, okay. I understand that they can breathe a bit. They can breathe due to through their skin. Okay, but I don't know. So Daniel's frog were under hibernation and they were breathing.
Yeah, normally
But
they're in the burrow. But as I said, they burrow in the sand. They have big grains. So
big grains of sand, so the oxygen could
goes down. You have lots of oxygen, 5 meters. So you can. Okay. I don't know if a human being can breathe there, but the frog they do. So there are two strategies in hibernation. You have an aerobic hibernation and
you have the aerobic one. Yes.
Yeah. So it's very important for us to know which one is happening, so you can direct and plan what you are going to measure, and then how to interpret your results. So it's very important to differentiate which case do you have. Okay. You know, I have a suggestion. You can. I mean, it's 10 years later, but you can.
Yeah, but we can do it.
Yeah, just do a simulation of the environment 10 years later. So you may know now if these animals were really in environmental hypoxia or not, because they can get hypoxia by the will. It's different. You have oxygen, but they decide not to breathe. So they will to become hypoxic, or they breathe very briefly. For example, some snails, yeah, they can become hypoxic because they want, yeah, but there's plenty of oxygen. So it's that nature is very variable. You can't take one example and then from one animal, one species, and think, oh, this is going to happen with my species, because nature has many choices, yeah. So you have to know which one you will get. I am getting very inspired. I am rewatching the series Dr. House. So I'm getting lots of inspirations. The way he, to pin down a disease and how to treat all the possibilities. It's the same thing in nature. Yeah. I'm talking too much. I have to let other people make questions.
Okay.
I'm sorry. I talk a lot. Let's people please ask questions.
Tell us how to redo this thing. I would think that we can put the animals in different soils of different size grain, and we can see if they become hypoxic or not, something like that. But let all the others speak, because yes, if they are questions.
Please, people, ask questions. We have talked a lot.
Probably we have covered with our discussion everyone.
Yeah, if people want to make questions about this thing of doing work in nature, so we can all discuss this. This is very important because Costa wants to redo this with real animals from really from nature, yeah, not a simulation anymore. So maybe people have questions about their own models. It's a good moment to ask questions. For example, I think Alexandra, she's trying to do some measurements. So we did some talking last year about doing some collection of muscles in nature. So this it just on a philosophy of work. You know, we have to embrace this philosophy and be prepared with all things that we have to be aware to measure when we go to nature. Because when you missed, then you cannot go there and remeasure. You just missed. For example, UV radiation for some animals is very important. For some, maybe not.
I agree, but hello everyone.
Oh.
Hi, Alessandra.
I agree with this philosophy, but I also to do these experiments to save probes and bring it to the lab. So I understand all the questions that are asked to laboratory experiments, but for example, for me now I understand that in laboratory work I am quite good, but in natural experiments I'm like a junior scientist, and I think that it's really difficult to for me to even to plan the experiment, especially if you are living in the area where there are no such fluctuations of the conditions, for example. I think it's really, and we here in Russia, we also have these probes, and this autumn I worked with them, and but I studied another effect. I studied the adrenergic response during the and it's dependent on seasonality. But for me, it's practically nearly impossible to have probes from, for example, winter samples when they are hibernated in nature. So I think that these problems are similar in every scientific group that try to organize natural experiment.
In places that are very cold with too much snow, so it's a big problem to find animals. Yeah. So the trick we did in our work, just to I'm not sure that is exactly what Alexander was saying, that we in the work we did in a semi-arid area of Brazil, it's quite kind of a desert. Yeah. But there was a farm there, a private farm, and so these physiologists were there for 10 years before we started to collaborate. So they knew the area very well. So they know, oh, this place here, if you dig, you find animals. So they knew exactly where the animals were. So they're true biologists. So the secret to do these experiments, you have to be partner with a zoologist from nature that works in nature, that knows the area we're going to, knows the animal. That's the only way, and you know the place. Then you can plan these experiments. That's the big secret. We just use our biochemic spirit, it won't work because we were not trained in nature. So for me, it was like a 10-year. More than 10 years of how we say hitting the head in the wall to learn, yeah, doing a lot of mistakes. So we will be pleased to share our mistakes and our positive experience So if people wants to try do something in nature, we'll be able to help, at least with advices. Yeah. It's really a challenge, but I think that's the big that's the big frontier. Yeah. There's so many papers are being published every year with lab simulations, many per year. So there is no more novelty. Yeah. So the frontier, I think, is nature now. See, is this mechanism happening in nature? Oh, okay. Just a sec. Oh, sorry. People, please ask questions. Thank you, Alexandra. I want to talk with you maybe tomorrow or Friday for some ideas. But people, please ask questions to Costa.
No one?
Yeah, I think today people are kind of shy. Yeah. Yeah. Okay, if we don't have more questions, so I'd like to. Okay.
Sorry.
Yeah.
Okay. I think that the most important this topic is to see what's happening in mitochondria. I don't know. Hosaska published their work about the mitochondrial affinity, oxygen affinity. We say hypoxia, but it's really hypoxia. Okay, we have a reduction of oxygen consumption, but the mitochondrial affinity of oxygen probably is high.
Yeah.
And even if we have a low oxygen consumption, in fact, we have a high hypoxia because that's the main cellular mechanism where we can see whether we have aerobic or anaerobic oxidation. I think that we can parallel these two processes. That's really very important. And of course, we can, I don't know who is working on all these animals, anyway, hibernation, hypoxia, etc. To see what's happening with tissue. We have plenty of mitochondria or not. It's a different working on white muscle than to red muscle or in the heart. Really. And of course, many times we get suggestions working in wrong tissues. Working on white muscle doesn't mean that the same strategy is for her heart or for the red muscle. Very different. And that's really a question so many years I have it in my mind about these strategies. Okay. And really, I don't know how we can associate all this mechanism with the post hypothesis. Really, we have the same responses in the heart, the white, red mass, etc. Where is the most potent this hypothesis, this mechanism? The white mass with low mitochondria, or in the red mass in the heart mass? I don't know. That's what I'm discussing at this point.
You hear me now? I think that from what I can remember, I think that in both the muscle and the heart, in this species, in these experiments, we had the similar results. We didn't have a tissue specificity of this kind that we see in other animals, probably due to the strength of overwintering and arousal, because the whole animal has to be aroused and it has to be metabolic active and stuff like that. I think that it would be a really nice idea to study what is happening, as you said, Marcelo, and also Vasily said, what's happening in anaerobic and aerobic oxidation, to see what happens in these two periods or before the animals go into torpor. I don't know if it's a good idea or not, but we can see how energetics are divided or not, maybe during the period of overwintering and arousal, and to see if there is oxidation, aerobic or anaerobic.
Yeah, it's a good point. Good point. Very important.
Yeah, but I would love to see what's happening before. I would love to see what's happening before animals go into overwintering. Because
if they have a preparation for
yes, yes. So what does this preparation consists of? I would love to see what's happening in the days before.
So is there a lot of papers on the preparation for hibernation, these animals, like the studies in bears, for example? There's a lot of work. They get fat, they accumulate a lot of fat. Yeah, they get
also
insulin resistant. Huh?
Also in squirrels.
Yeah. Squirrels is a lot of work. I mean, in this species of frog.
No. I don't think that many papers have been published regarding this type of hibernation and what is happening in this species of frogs. I don't know if I'm correct. Vasilis can correct me.
Another point is that we studied the enzymatic activities of antioxidant enzymes. But what about the gene expression? In many cases, we have the expression early, before seeing that we have increased or not the enzymatic activities. I'm saying that it's a high time to see about this hypothesis, about the thermal sensing. I mean that the cells and animals are sensing earlier what's about after 2 or 3 months. And that's why they are prepared. That's why we must associate both gene expression and enzymatic activities or the other metabolic processes. Okay, because we can see that the gene expressions are taken earlier, even before animals enter hibernate. That's a very high.
You have to consider covalent modifications of these enzymes, these target enzymes, because I think it's a combination of gene expression and phosphorylation, acetylation, and many other things that happen with these enzymes. So it's combination of both things. And microRNAs too, they probably play a very important role, to inhibit a lot of stuff, and let go other ones,
Because we have found how that in muscles, for example, before we found that the gene expression of the oxidant enzymes take place at 22 degrees Celsius, but we have the mortality. Mortality is taken after 26, 27 degrees in muscles. So why these genes are expressed earlier. Really, it's for me, it's a point to see when this process starts.
Yeah.
And it's very important for practical reasons. I mean, for management of some populations that are, because I'm reading so many papers. Okay, this animal died at 28, 30 degrees, etc. But what about earlier? What's happening earlier?
Yeah, it's very important to see the whole cycle. Yeah.
Yeah, the time
Before-
course. The time course, all these processes. The time course. Okay.
Yeah. Okay, thank you. So any more questions? People, don't get shy. So Sergey, I don't think it's Sergey's here. He works with, in a very cold place with frogs, and he's doing work with frog hibernation. So you guys might want to talk. Yeah. Is Sergey here?
No, I don't think he is.
Okay. Yeah, that's too bad. He's doing a lot of work in metabolomics of hibernating animals. He's doing earthworms, frogs, and other groups.
I would love to see also what's happening concerning transcriptomics during this kind of periods, because you have these groups of genes. You don't have to study one gene or a second gene, but you study the genes that are associated with oxidative stress or the cellular response, and you can see what's going up, what's going down. So it would be really nice also to do this thing about transcriptomics.
Oh yeah. Sure. Yeah. Okay. So do you have questions, or we are going to have lunch?
To take the picture.
Yeah. Let's open the. Where is my video? Who can take a picture? I am using my wife's computer, so I'm not very good in this computer.
I can take a screenshot and send it to all of us if we open the cameras.
Yeah. So people, please open your cameras.
3 more.
Yeah. I think the next seminar will be by Professor Alexandra from Sebastopol. I think she's the next. Okay, did you take a picture?
Yeah, I took 3. We can smile all,
good.
and I can take another one. So yeah.
Great. That's great. So thank you, Costa. It was very nice. We have a big discussion. So so I have a suggestion that we could open for students to present small studies. So this study, you were a student at that time, yeah? A PhD student or postdoctoral fellow in 2013?
This was as a postdoc.
Postdoctoral, yeah.
Yes.
So it will be nice to have students presenting even unpublished results. Yeah.
Mm-hmm.
For discussion, it will be very interesting. Yeah. Okay. So thank you very much. So see you next time.
Next meeting.
Okay.
Thank you very much for having me.
Marcelo, thanks a lot for
Thank you.
organizing this meeting.
Thank you.
Really.
Yeah.
Thank you very much. Thank you very much for very interesting presentation.
Yeah, that's good. Thank you. Nice to hear you, Alexandra.
Where is it? What the weather is this over there in Brazil? The summer, spring, or I don't know.
Here.
Yeah.
Oh, we are finishing summer.
This summer.
So you're
Finishing
going into
summer.
autumn. Okay.
Still very, very hot here.
Very hot.
Very hot here.
In Brazil.
Yeah, and a lot of rain here.
Okay. Have a nice
Okay,
day.
bye bye, people.
Okay,
See you.
bye-bye.
See you next
Bye-bye.
time.
Bye bye.
Thank you
Bye-bye.
again,
Thank you very
Constance.
much.
Thank you
Yeah,
again.
yeah. Bye bye.